the right dose differentiates a poison from a remedy. Okay, this is told by Paracelsus, father of toxicology. So anything, benda apa pun, kalau dimakan, kalau dia melebihi had dia akan menjadi poison. There's a proverb lah, in Tamil there's a proverb, they said whatever you eat in excess is a poison. Okay, so you have to know the dosage. It's not just food, it's not just medicine, it's also the food. Okay, if too much, it will be bad for your body. Okay. Okay. The ability of a substance to cause toxicity is the ability of a substance to cause injury to biological tissue. So in medical term or in a science term, toxicity, that is the meaning lah. Okay, if the substance ability dia kalau dia increase to certain level, it can cause injury to the biological cells. Okay, toxicity could be caused by chemical stimuli. Chemical stimuli means poison lah. Mostly the chemical poisons is called chemical stimuli. Exposure to other cells. Okay, you're exposed to NK or T cells. Okay, sometimes this even in our own body can the exposure to other cells can cause toxicity. Sometimes our body have a mechanism that fight its own body ataupun its own cells ataupun other cells kan yang datang dari luar, like bacteria yang infect kita or anything. The body will consider that as a foreign items. It will attack, it will kill it. But if it's overdose and then body's phytosystem kan, phagocytosis tak boleh attack dia balik, then it is also considered poison. Okay, physical environmental condition like radiation exposure. Okay, so if you have higher radiation, like UV is high, now our UV, we are protected by layers of UV protection. Then so if it is known that not there, then we might get what is this? We might okay, sorry students. Okay, radiation exposure, like okay, X-ray radiation, it's also radiation kan, but that kind of radiation is a very small radiation. If you go for some kind of radiotherapies due to cancers and so on, then it is a very big radiation lah. It can also cause toxicity to the cell. Okay, let's say my father is a cancer patient, he's going for radiotherapy. That radiotherapy ni dia akan bagikan radiation to his cells, to his cancer cells, to kill the cancer cells. But it also kills the cell around the cancer cells, the normal cells as well. That is also toxic. Okay, temperature or extreme pressure. Okay, some of them they die due to climbing Mount Everest. Halfway they cannot stand, and some of them die because the pressure is different. Okay, this is called this also causes toxicity to your body. Okay, why is it important to measure toxicity? To establish a dose response curve. There will be a curve later you will see where you can check okay, at what point is it the toxicity or the dosage of a medicine starts to work. At what point it is made 50%, at what point it's saturated, and if over that, it might cause poison, might be poison. Okay. To ensure safety of new chemicals for use such as pesticides, drugs, or food additives. Okay, so new chemicals you have to test that. You have to test toxicity so that it macam you sembur racun serangga ke apa kan. Okay, so you must know it is not poisonous to the plant. It is not poisonous to even human who are using it on the plants. Okay, drugs or food additives. Okay, so food additives pun kalau too much it can be poisonous. Okay, before they are registered for general use in industry or doctor's clinic. Okay, drugs kat sini, it's not the drugs like yang being misused lah. That one, the one that is being misused is of course it's causing toxicity to the body. Okay, that's why they say don't misuse the drugs. These drugs is made by medicines. Okay, medicine is also drugs. Okay, anyone can tell me what is the drug that is consumed by more than half of the world's population, but and it is legal? Anyone knows the answer? Hello.
Panadol.
Huh?
Panadol.
No. Panadol, it's not considered it's consumed by half of the world. It's a drug that's like more, okay, 70% of world population consume that. It is a drug and it is a legal drug. I mean like Panadol pun kadang-kadang ada orang tak boleh ambil. This one you no need doctor's prescription ke, tak perlu. Any guess? Maybe you also took it today morning.
Marijuana?
Huh?
Marijuana.
Marijuana tak ada lah. Tak legalize lagi. Malaysia pun tak legalize. Coffee. Caffeine in coffee. Coffee is a kind of drug which it stimulates a lot of things and it's legalized. And even if you drink it a lot, there will be adverse effect kan. Macam tak boleh tidur lah, sakit kepala lah. So that is actually coffee. Caffeine is a drug. I mean like it's a component of a big drug, but it's like we all consume. You don't need prescription and so on. Okay. Okay, we continue. To establish the mode of action or mechanism for the toxic effect that may have been seen in other studies lah. So they want to confirm other studies. Okay, what's the mechanism for the toxic effect? Okay. To produce epidemiological studies to explain observation in population, for instance. So a long investigation into association of smoking with lung. Okay, earlier last time, like in you were also not born, even I was also not born, in 50s, 40s, smoking was not considered as something bad. People doesn't know it has a harmful effect until one day they started to do the research and they realized smoking is the bad part of it. Okay, so it's also a drug. So you need to study. This is a ongoing process. Like smoking is something like it is a chronic disease. We call that it causes chronic disease. Like it slowly manipulate your lungs, your so many things lah. Okay, so it's a long thing. So you have to observe it for a long time. To validate new methods of testing or investigation, particularly those conducted in vitro rather than in animals. So to validate what they have, okay, first animal test is the best thing lah. Like some animals like chimpanzee, monkeys, or like rat or rabbit that are used is the best test to decide like, oh, how much is it toxic? So they try to relate it to human because you cannot test toxicity or dosage on human being. Okay, it's against the ethical right. Now, animals testing is also being an issue. A lot of people are questioning about that. So the first, before they start with animal tests, they will do in vitro tests. Okay, they will do outside of living organism lah. Maybe they will take one kind of organs or anything. They will try to test that. That is called new method. So once they have tested in vitro, new method of testing, particularly those conducted in vitro rather than in animals. So once if they have tested in vitro, they think it's correct, they start to do in animals and then humans. Okay, they will test it. Okay, like how our COVID vaccines were done and so on, like they have testing. Even humans also, they have a batch that used to test this effectiveness of this vaccine. Okay. Okay, the two basic principles guiding toxicity tests in animal. To check the effect of the test substance on laboratory animal and its direct toxic effect on human. Okay, so first, they test the effect of the test substance, the compound, the drugs, on animal, then only they do it and it's on human. And they check also the direct toxicity effect on human. Exposure of laboratory animals to high doses in order to evaluate its possible hazard on humans. So they have to test it on animals. So in human, you cannot like try to test, or you cannot, as I told you, you cannot give high. I mean, it might be a high dose, it might kill them. In animals, we can do that. Okay, there's still a way you can try to check on animals. Okay, during Nazis during World War One, when Germany was in power, when they took over a lot of nations in Europe, okay, and East Europe and so on, what they did is they did this. They do a lot of experiments using human themselves, whoever they have captured, the Polish, the Jewish, and so on. Okay, that was the main reason it was banned. Okay, that they are exposed to much lower doses. Okay. Toxicity can result from adverse cellular, biochemical, or macromolecular changes. Examples are um, so toxicity can cause bad things is like it can cause cell replacement, such as fibrosis, damage on the enzyme system in the body. Okay, enzyme system is very important. Even for gene regulation, enzyme system is very important. Disruption of protein synthesis, disrupt the protein production of reactive chemicals in cells. Okay, then it causes DNA damage. Okay, so all the adverse bad effect of protein synthesis. Okay, measure of exposure. Exposure to poisons can be intentional or unintentional. So you can be exposed to poison intentionally or intentionally. The effects of exposure to poison vary with the amount of exposure. Uh, the effects of exposure to poison vary with the amount of exposure, the dose lah, how much it was um, exposed lah. Okay, so dose, it depends on the dose. So if you go to a hospital, if they say take 2 pills of these medicines every day, every day once, two of these pills. Okay, maybe they say 500, 100 UG and so on, 1000 UG and so on. You go and take 4. It means the dosage is high. You are exposing yourself to toxicity. This is the issue when you go to like you use traditional herbs or traditional medicines and so on. Can sometimes they ask you like traditional medicine practitioner, they doesn't know about dosage. Sometimes it's over for your body. Terlalu banyak, macam you dah ada diabetes, lepas tu you pergi jumpa perubatan tradisional for something other than that. They don't know about your diabetes, so they subscribe you something lah, ask you to eat. Mungkin ubat yang dia suruh you makan tu, dia punya dose tu adalah tinggi kerana you are diabetes patient, it might increase your diabetes. So that is very important. Okay, that's why they said go to a proper certified practitioners. Okay. Dose, the amount of substance administered at one time. That is what the meaning of dose. Or to characterize the exposure to xenobiotics. Okay, number of doses, frequency, total time period of the treatment. Example, you will only be administered 650 microgram Tylenol as a single dose, ataupun 500 milligram of penicillin. Macam Panadol semua, you eat 5000 mg every 8 hours for 10 days, then it might work. Okay, same goes to certain medicine like vitamin C kan. Vitamin C is something you can eat every day, but it won't be poisonous because when the amount reaches more than certain milligram, the body will buang benda tu. Okay, you will throw, body won't use. Okay, so certain medicine like Panadol kan, if you're sick, if you take less than 300 milligram in 8 hours, it doesn't work. The dosage tu tak cukup untuk trigger the treatment pun. Okay, in the curve. So it must have certain percentage to start the treatment. Okay, to start the effectiveness. Okay. Uh, Dosage is expressed in terms of quantity administered, okay, per unit of weight lah. Like how many kg, like 500, how many milligram, 500 milligram per kg ataupun 500 microgram per kg. Sometimes the kg can be our body weight, like for 80 kilogram, for human with the 80 kilogram, how much those how much milligram of medicine that can be given. Okay. ppm, parts. Ppm is I think if you had chemistry classes, then you should know ppm is part per million, part per billion, this one. Okay. Skin surface, by a milligram, kalau it's a skin surface, if you are like uh, sapu ubat kan, macam orally administrated, bukan oral, non oral, okay, non orally administrated. So it is like milligram per centimeter square, berapa besar tempat tu kan, okay, to check the dosage. Okay. This is what I was telling about this dose response curve. It describes the effect on an organism caused by differing level of exposure or dose level lah. Okay, caused by differing level of exposure, how much you can expose. Okay, those levels are usually expressed in milligram per kg body weight. Just now I was telling you kan, how many milligram you can give to a person of this much of body weight. Okay, that's how they check the dosage. Okay, of the test animal lah, for solids. Okay, they used to check those levels in animals, as I told you, animal as a test materials. Okay, if it's a solid medicine lah, kalau it's like a vapor kind of medicine like essential oils, or they use milligram per meter cube, macam area per meter cube, or parts per million. Okay, this level can be plotted on a graph against a response. Dose response curve is a valuable tool to understand the level at which substances begin to exert adverse effect. Okay, kena dia nak tahu macam dia tengok curve tu dekat mana curve tu macam boleh give adverse effect, bad effect. Okay, exert adverse effect and the degree of harm expected at various level. That is very important. Okay, orang yang buat dosage study, toxicity studies, they are well familiar or aware of this toxicity curve. Okay. Dose response curve shows a number of points including the no effect level. No effect malah, if you give this much, nanti I tunjuk you the graph. There's no effect. You have to increase the dosage. Kalau you give less than that, there won't be any effect lah. Okay, no effect occurs or not detectable. The threshold dose of a substance. Threshold means the level at which the effect starts to occur. Bila dia mula tunjuk response. Okay. The level at which the effect occurs in a set percentage or all of the test organism. So like 50% of the test organism is responding to the dosage given. So there's a term for that lah. Okay. Okay, important assumption. There's almost always a dose below which no response occurs or can be measured, as I told you just now lah kan. Okay, because the drugs tak ada effect pun at that dosage, very low dosage. Okay, once a maximum response is reached, any further increase in the dosage will not result in any increase of treatments. Okay, it won't give any benefit lah. Okay. Okay, this is a hypothetical dose response curve. Okay. Dose response graph typically have the dose on the x-axis and the measured effect. Okay, this is the concentration. Okay, and then this is the response. Okay, plotting y-axis is the response lah, how the body responds, how the disease responds. Okay, plotting the logarithm of the concentration generally result in sigmoidal plots as shown on the left. Okay, so this is the plot kan. So A, okay, this is where if the concentration below this, there's no any effect, okay, from the drug. So this is where the point starts. This is called the potency lah. The main parameter is the potency. The position of the curve along the x-axis is this is the potency. This is where the effect starts lah. A. B is a maximum efficiency. The greatest response attainable is B. After this, there's no any response. I mean, like, it will be stagnant lah. You ambil lebih dari ni mungkin akan jadi poisonous, and it will be like the great response already start. There's no use to take further. Slope change in response per unit. And half this is the slope lah, and half of the concentration is the C lah. This point C. So this is half of the concentration. You see, usually this 50% is very important in the studies. Okay. Like I said, vitamin C kan, if you take more than this concentration, tak ada maknanya. Our body will throw it out, like it won't be toxic to our body. But certain things, it will be toxic if it's too much. Okay. This is the comparison of dose response curve lah. Like for the let's say this is A, this is drug A kan. So dia you give them this much, okay, from this point yang I tunjuk mula ada effectiveness. B, it takes more than that of concentration. C is higher. Okay, let's say one example I can give is we have variety of vaccines kan. Dalam for COVID kan, that day we have tiba-tiba I boleh lupa vaksin. Pfizer, we have AstraZeneca, we have Sinovac. So every different vaccine yang produce, dia punya concentration yang mereka start to give is different because of this lah. Okay, it take effect at the different dosage. Okay, this is very important. You have to know this anyway. Particular points of dose response curve include LD50, lethal dose, 50%, dose that kills 50% of the test population. Okay, so LD50 means dose. If you give this dose, it might kill 50% of the test population. So if you test with 10 different test population maksud dia test population, like if you want to kill a bacteria, okay, if you give if you have like 100 milligram of bacteria ataupun 100 population lah, hundreds of bacteria, so if you can kill 50 of it, it is called lethal dose, LD50. Okay, so that's dose that kills 50% of test population bukan manusia lah kan, bukan dia test on kita, benda tu akan bunuh 50% start. Dia test population of the bad things lah, of the organism that causing the toxicity, maybe the bacteria or fungus and so on. LC50 is concentration that kills 50% of the population. Like this is the dose, this is the concentration. Maybe LC50 will come across maybe in the lab tomorrow. Okay, so this much of concentration of the compound will kill, okay, the animals or something like that lah. I will kill the bacteria. Okay, TD50, those that causes a particular effect of the test population. Okay, so TD50 it cause particular effect lah, 50% also. TC50, concentration that causes particular effect, concentration of medicine. Okay, that causes particular effect in the test population. The lower the value, okay, kalau it says TC50 dia adalah 30% je, 30 je. So the more toxic the chemical, because kalau dia kata TC30 compared to TC60, TC30 tu lagi toxic. Maksudnya apa? Dalam 30 TC50 dia adalah 30 mg. Dia kata 30 mg is enough to achieve TC50. Okay, another medicine it says 60 mg is enough to reach TC50. Maksud dia 30 mg tu lagi toxic. Pasal apa? Dalam 30 mg dia boleh bunuh half of the bad bacteria punya population. Tapi yang lagi satu tu dalam 60 mg baru dia boleh bunuh. So it is like the 30 tu lagi toxic lah maksud dia. Okay. Okay, this figure illustrates how an LD50 of 20 milligram is derived. Okay, this is the dosage in milligram. This is the response curve. So when they do the response curve, they do it until here. Dia dah macam that flat face kan. That face ni lepas ni you take the medicine pun dia takkan naik. So until here, so they check the what's the 50% response lah. So at 20 milligram of the dosage, it reaches LD 50. Okay, half of the effect lah. Okay, types of toxicity. We have acute toxicity. Okay, effects usually within 24 to 72 hours after exposure. However, should occur within 14 days of exposure. So acute and chronic disease pun kan, they are acute disease, chronic disease. Acute kalau dia tiba-tiba macam it affects now, tiba-tiba it give effect now itself, macam very very fast, very very immediate. That's why they said like 24 acute toxicity pun 24 to 72 hours. Okay, kalau chronic it's like a slow and steady. It's toxic but dia punya effect the effect is slow lah. It moves slowly. Okay. Effects usually within 24 to 72 hours after exposure. However, should occur within 14 days lah, 14 hari. Chronic effects manifest several weeks, months or years after exposure. So it take time lah, several weeks, several months or several years, okay, for it to show effectiveness. Like one example, it can be like there's a like diabetes is a chronic disease and the toxicity pun daripada sugar and it takes time. Diabetes, it won't develop like it cause like sugar tu macam tak adalah tiba-tiba dia bagi effect kan. So slowly daripada a small pre-diabetes and then it moves like 10 years and then it cause the disease. Okay. Kalau acute tiba-tiba terminum poison, it cause acute poisoning. Okay. Local toxicity means action of toxic substance on specific area of contact. Okay, on the skin, on the mucous membranes, on the eyes, on the throat. Okay, that's the local lah. It's like the one part of a body. Okay, local toxicity. Systemic effect or action is distributed throughout the body. Like say inhalation of chloroform. So that's a chloroform ataupun mercury chloride. These are all like very very poisonous. So when you inhale, it will be systemic. If you inhale through brain, it can affect the whole body. Okay. Carcinogen, I think you all know that carcinogen is the thing that causes cancer, kan? The compound, okay, substance that will induce malignant tumor. Tumor means malignant tumor means cancerous cells lah in a person following reasonable exposure. So too much of exposure to radiation, to chemicals, and so on can cause cancer as well. Mutagen, okay, affects the genetic system. It causes mutasis lah. Mutasis, okay, mutasis, okay. Affects the genetic system of exposed people, animals, and plants, and may cause cancer or undesirable mutation, mutasis mutation in future generation, like the bombing of nuclear to Hiroshima and Nagasaki. Okay, the nuclear bombs will cause a mutation to the people, and then it causes like in the X chromosome, Y chromosome, the next generation. Okay, this causes a lot of issues. Okay. Teratogen is a compound that affects the fetus to the mother, causing the death or significant deformities. Okay, it's from the mother actually. Okay. Okay. So that's half of the lecture. So we have another half. I think we can finish it. Anyway, any questions so far? Students, okay ke? So am I too fast?
Okay. Okay, Doctor.
Okay.
Yeah, faham ah. Okay, not too hard lah if you go and read. Okay. Anyway, exam question I dah prepare final. So every topics of my lecture is coming out. So every topics maksud dia my lecture lah, Dr. Dr. Linda punya tak ada. Okay, the 7 weeks. Okay, that's the tips I can give you. Every topics ada question. We have 6 short answer question, 3 essays. Okay, it's a 2 hours exam. So be ready. Okay, and now can we take like a 3 minutes break? Okay, then I'll be back. Okay, pukul 10 you orang ada kelas kan?
Okay.
Pukul 10 ada kelas ke?
Ya,
Ada.
Ada.
Okay, okay. Ada, okay, okay. Take 3 minutes break lah, I'll be back. Okay. All of you are here? Okay, so we shall continue. Cell physiology. Okay, treating cells with the cytotoxic compound can result in a variety of cell fates. Okay, so when you treat them with cytotoxic compound, okay, toxic compound, like a lot of things can happen. Okay, the cells may undergo necrosis ataupun the accidental cell death. Okay, they lose membrane integrity and die rapidly as a result of cell lysis. Okay, they lose the integrity of the membrane and they will die rapidly as a result of cell lysis. Okay, cell akan lyse kalau dia akan break out. Okay, accidental cell death ataupun programmed cell death, apoptosis. The cells can stop actively growing and dividing. Okay, it stops growing, it is killing himself. Apoptosis means macam the cell akan mati by itself lah. Okay, it's like not because it's lose integrity ke apa. Okay, it stop growing and dividing, a decrease in cell viability. Okay, or the cells can activate a genetic program of controlled cell death. Okay, any a decrease in cell viability means decrease means kurangkan viability means kurangkan kehidupan lah. Viable, it's not viable means it's not alive lah. A decrease ataupun it will activate a gene program that will control a cell. Dia akan macam bunuh diri lah. Cell macam tu dia akan, it will kill. Okay, it can also happen when your cells are old and so on. This thing can happen. Okay, so this is two kind of things lah. Yang ni macam bunuh diri, yang ni macam kena toxicity tu dia akan bunuh diri, ataupun yang ni dia kena toxicity tu sel tu akan kehilangan kekenyalan dia ataupun integrity dia and so on, dia akan mati juga. Okay. Okay, necrosis. How it happens? Necrosis typically exhibit rapid swelling. It will be a swell. It lose membrane integrity. It shut down the metabolism, so it won't have any metabolism happening there. Release their content into the environment, so there won't be metabolism. Dia punya content dalam badan dia punya subcells is all released outside. Cells that undergo rapid necrosis in vitro do not have sufficient time or energy to activate apoptosis. So, dia tak boleh activate apoptosis dalam in vitro and will not express apoptotic markers. So, it will die macam tu lah. Okay. Apoptosis pula, it's a well-defined cytological and molecular events. Okay, it's a well-defined, like it's a process that done slowly by the cell itself. So, macam bunuh diri lah, it plants it's this thing. It change in the refractive index of the cells. The cytoplasm will shrink, the nuclei will condense, okay? And then the DNA will be cleavage into regularly sized fragments. Cells in culture that are undergoing apoptosis eventually undergo secondary necrosis. They will shut down metabolism, lose membrane integrity, and it will lyse. Okay, that is how it's a difference lah. Okay, it's like apoptosis is more like a programmed, well-programmed kind of cell death. This is suddenly happening due to the dosage. Both also can be done due to the dosage, but this is more planned lah. Okay, like cells. Okay. Okay, assessing cell membrane integrity is one of the most common ways to measure cell viability. Okay, and cytotoxic effect. Okay, you check the cell punya membrane integrity. So to check the cell viability, kehidupan cell lah. Okay, lifetime of a cell and the cytotoxic effects. Compound that have cytotoxic effect often compromise cell membrane integrity. So it has the compounds ataupun medicines ataupun any compounds that you give to the cell that have cytotoxic effect, it will change the membrane punya integrity. Okay. Vital dyes, important dyes such as tryptophan blue or propydium iodide are normally excluded from the inside of healthy cells. Okay. However, if the cell membrane has been compromised, they freely cross the membrane and stain the intracellular components. So these are kind of dyes that you can use. Okay, later we'll check that. Toxicity testing. Toxicity testing on animals remains the main approach. Potentially controversial issues. Progress has been made to replace animal tests, reduce numbers used, and refine testing methods to reduce suffering. So this is controversial issues lah. Okay, on animal. Okay. So they are now using a lot of in vitro tests, like animal tests they're using, but they're at the last level. Okay, most toxicity tests use rodents. Okay, rodents example breed rats lah. Kan dia tak ambil lah apa tikus jalanan ke apa kan, dia breed the breeded mice. Okay, rat is a bigger version lah, mice is a smaller version. Okay, guinea pigs or rabbits. Tapi it's all breeded lah for this reason. Okay, other approaches use cells or tissue in culture. So they take out the tissue, they do it in vitro. Okay, test tubes or petri dishes lah, also known as in vitro. Okay, in order to determine the cell death caused by various mechanisms, there's a need for cytotoxicity and cell viability assays, which are cheap, reliable, and reproducible. This assay can be classified according to measurement types of endpoints, color changes, fluorescence, luminescence, and so on. Okay, these are type of different type of assays that can be used to check cytotoxicity. Okay, there's a dye exclusion test, tryptin blue, eosine, congorate. Okay, colorimetric assays, MTT assays, MTS assay, AZTT assay, LDH assay. There's also fluorometric assays, alarm blue assay, and CFDA AM assay. Okay, luminometric assays. Okay, and so on. Okay. Okay, I'll go through one of the assays. Tryptin blue assay. Okay. Dalam tryptin blue assays ni, live cells possess intact cell membrane, intact lah. Okay, good cell intact, very compressed, that exclude this dye, whereas dead cells do not. Okay. So if you dye the cells, you want to check which is dead or alive, the blue dye will dye the dead cells. The live cells won't be dead. So as you can see the white part here. Okay. Cell suspension is mixed with dye and then visually examined. Viable cells, yang viable means yang alive lah, viable cells will have a clear cytoplasm, whereas the dead cells will have a blue cytoplasm. Okay. Counting is generally done using hemocytometer. Hemocytometer means I think you have done, maybe you will done in future in maybe plant pathology class and so on. So it is like you know, there's like a you think about microscope, there's a slide yang ada macam kotak-kotak petak, so you can count one by one. Okay. Based on the principle that the living cell membrane has the ability to prevent the entry of the dye. Okay, living cells, dia tak bagi dye to masuk. It's integrated lah, hence they remain unstained and can be easily distinguished from the dead cells which take the dye. So how do they do that? Okay, if you have never done molecular biology, or you will do this lah. Okay, this is centrifuge tube kita panggil dia. So kita masukkan the cell suspension yang ada cell tu dengan dia punya buffer or anything. So we will spin them in high speed. When you spin them, cell akan ke bawah, supernatant akan ke atas, air ke atas, cell aja ada kat bawah. So you can throw the supernatant and then the cell pellet in PBS. PBS can be the buffer lah, buffer, and they add the dye tryptin blue. Okay, and then they incubate for 25 degrees for 3 minutes. Then they put in the hemocytometer, and then they check, is it viable or apoptotic. Okay, kalau apoptotic dah matilah, it's stained. Viable, dia tak distinct. Okay. And okay, so I'll go through another one. Okay, region used in calorimetric assays develop a color in response to viability of the cells. They evaluate metabolic activity of the cells. So kalau calorimetric assay, dia akan you use this method, it will color the living cells. Okay, it's easy to perform, comparably economical. For example, this one people use a lot. Okay, in if you do studies with Dr. Mai, she does a lot of MTT assays. Okay, so MTT assay is 3,4,5 dimethyl 2-thiazoyl 2,5-diphenyl 2H tetrazolium bromide assay. Okay, measures cell viability through determination of mitochondrial function of cells by measuring activity of mitochondrial enzymes such as succinate dehydrogenase. So if a cell is alive, the mitochondrion must be alive, must be working. Kena mitochondrion yang produce energy. So they are trying to dye the mitochondrion. Okay, enzyme dalam mitochondrion tu. Okay, MTT or closed dyes, call it MTS, ZTT, are reduced to purple formation. Once dia ada enzyme, dia bertindak dengan enzyme, dia akan tukar warna. Product can be quantified by light can even quantify how alive it is through by light absorbance at a specific wavelength. Okay, maybe you can use a fluorometer. Okay, MTT. Formazan, which is formed, is purple colored water insoluble product that is largely impermeable to cell membranes. Okay, thus resulting in the accumulation between the healthy cells, which is solubilized by adding the MSO. This is one of the method. Okay, this is a cell plate. Okay, for this is a micro acid plate lah. Okay, they call pile slate. It has 96 wells. You incubate the cells for 24 hours. You add the MTT solution. Incubate for 4 hours. MTT solution. After adding DMSO, cover the plate, leave the plate in the dark for 2 hours, and then you check using spectrophotometer. So you can see how much is it alive and so on. Okay. Why they have different colors? They use different amount of dye in the assay. Okay, so they will check which has the highest absorbance and so on. Okay. I think this essays you can read yourself. Okay. Prediction. The highly important topic is the prediction of cytotoxicity of chemical compound based on previous measurement, i.e. in silico testing. In silico means, okay, you compare the cytotoxicity of the previous measurements. Kan? So you don't need every time to test. Now we have data online mostly for many things. So in silico means like using bioinformatics or using data yang ada dalam komputer type of toxicity assessment that uses computational method to analyze, stimulate, visualize, or predict the toxicity of chemicals. So computational method lah. For this purpose, many QSAR and ritual screening method have been suggested. Okay, so undergoing and future as well. Okay, you don't run everything using living organisms. Okay, in silico toxicity prediction. Experts rule based on example direct QSAR model. So I'm not an expert on this part anyway. So in silico, it more to like Dr. Hidayah and Dr. Mai's combination lah. Cannot replicate complicated interaction in the whole system. So example in cancer, chemotherapy as a treatment of cancer. Chemotherapy means this is a chemical treatment for the cancer, often relies on the ability of cytotoxic agent to kill or damage cells which are reproducing. Okay, cytotoxic agent lah, it use compounds. This preferentially rapidly cells. You'll target the rapidly dividing cells. So this is example is in cancer. Okay, that's it for today. Any questions? Links? Questions? Question. Dengar tak? That's done for your class today. So I will deposit this in your Italim, and then I also will deposit your your lab manual dalam Italim. Okay? So that's it. Thank you for today. Bye.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.